Good morning, ladies and gentlemen. We are requesting just a few minutes of your time. It is now 12, but we're just going to wait 2 minutes or so for new joiners to come in. Okay, once again, good morning, ladies and gentlemen. We are here today as part of our National Science Month commemorations, and we welcome you to our webinar today. We have reached the stage of our National Science Month where we're well into our block theme around environmental and conservation management. This is one of the key themes for National Science Month, as most of the work we do in the science, environment, technology space is around conservation management and protecting our environment. So we are very happy that we have a esteemed researcher in the conservation management space with us today. We look at the fact that South Africa is the third most biologically diverse country in the world. Approximately 10% of the entire world's plant species, 7% of the world's reptiles, birds, and mammals, and almost 15% of the world's coastal marine species are actually found in South Africa. So this is even more reason why we as South Africans should step up when it comes to our environment and conserving our environment. We live in a very urbanized world. I think the statistics are up to almost 70% of South Africans actually live in urban areas. So this means we're quite often out of touch with our environment. For example, how far are we from our food sources today in today's world? We go into a shop, it's a nice, clean environment in a supermarket, and we go up to a shelf. We select food that has been sourced for us, has been cleaned for us, has been packaged for us. And it's also very convenient. But the further away we are from our food sources, the more out of touch we are with the health of our environmental system. And this is why today's topic is so crucial. Imagine walking into your favorite supermarket and looking for your favorite freshwater fish and not finding it. You'd probably think, okay, they're out of stock, but weeks and months later, you're still not finding it. This is a key example of how, when we are out of touch with what's happening in our world, we are less likely to see the challenges, the problems, the negative effects that are happening in our environment. So today we have with us Dr. Matthew Burnett. He's the principal scientist at the Institute of National Resources. He's also an honorary researcher at the University of KwaZulu-Natal. And so he is an expert in what makes our freshwater systems tick. He focuses on what is called fish telemetry. So, not being a scientist myself, this was the first time I came across this word, fish telemetry, when I was asked to facilitate the session today. So I did a little bit of Googling just so that I'm not completely ignorant. And fish telemetry has to do with tracking the movements and the behavior of fish or aquatic animals in general, and also monitoring their habitat. So today, Dr. Burnett is going to talk to us about understanding our freshwater ecosystem a bit better, monitoring what's going on in our environment, and being able to know in advance when there are problems in our freshwater systems. So fish telemetry has to do with the tracking of the of fish as they move between, for example, their breeding areas and their feeding areas. It also looks at the actual habitat and whether the habitat is still conducive for the growth of our species, aquatic animals in the water that we use. So conservation then is a very big sign of when there are problems and when we need to, as humans, step in. So the work that Dr. Burnett does is the work that tells us how healthy our system is, how much of our aquatic animals are still healthy, enough for us to be able to use it as a resource for food, for also in terms of having a safe environment for leisure activities when it comes to water. So, Dr. Burnett, I didn't want to take up too much of an introduction. You are our expert. I'm going to hand over to you to take us through your research, and especially, I know you mentioned that there's some breaking news as well, coming out of the research that's happening. So please take us through.
Yeah, thanks, Lynette, for the introduction and the opportunity to share the work that we've been involved in. And yeah, it's nice to be able to present it here today. And yeah, like you mentioned, so fish telemetry is a field that enables us to study fish behavior quite adequately. And that's really what my talk is going to be about today is where we are as South Africa in using fish telemetry techniques to understand fish behavior and also how that relates to how we manage our water resources. So generally, how I introduce the topic is what you see in the background here is a nice example of what a river should look like. Not many of our rivers look like this anymore. Um, and um, and yeah, so maybe just an outline is how is we're gonna kind of touch on why fish behavior matters and why it's important, and then how we can measure fish behavior and relate it to freshwater management, and then also just two sections here on two kind of techniques that are used to measure fish behavior and how they can be applied. The first one is a locally developed fish tracking system, and then section two is monitoring fishways, um, and which is kind of like a first for South Africa and likely to be a first for Africa. And then just a short note on where to from here in using fish behavior to manage our freshwater systems. So back to the riverscape that I've got in the background here. When we look at fish behavior, we look at the river and what a fish needs in order to survive. And if we were to look at this landscape and put a bird in it, you can see that the bird is able to fly around. There wouldn't be much barriers or obstructions to its pathway through the landscape. If we were to then put a mammal, you might find that a rabbit on the southern bank might not be able to move to the northern bank because the river starts to obstruct its path to access different habitats. And then you start looking inside the river with the macroinvertebrates, the crawlies. They can generally move up and down quite freely and over in and out the water at times. And then you throw in another element in terms of not just that individual's behavior, but he's looking for other crabs to socialize with and coexist with. And so there's then this linkage between not just the habitats that they need, but also where they can find other individuals. And then you throw fish in there, and fish often are very much restricted to the water column and the water place and space. So as soon as there's an obstruction, a breakage between the water columns, you'll find that that system is no longer connected and becomes disconnected. And so what you find then is, not to go into too much detail now, but one has to aid the fish in terms of maintaining that connectivity in a system so they can perform their biological functionings. And then, yeah, just ecosystem management is looking at all these elements to make sure that all the biota in the system is managed for adequately. So as soon as we start to change this riverscape, are we catering for the biological organisms in that riverscape that allows us to, or allows those species to persist within it. So on the back of that, if we were to bring up this image that I've got, the question then is, we've now changed the riverscape by building a weir to abstract water, which is much needed in South Africa. But with that, we start to ask the questions, okay, so if a river downstream here needs to get upstream, what is its pathway to do that? Can it do that? What mechanisms have we put in place in this structure for them to do that? Here's another example of a in-stream structure. Again, how is fish going to move upstream downstream to upstream, but then also downstream, upstream to downstream. And so these questions start to become important. If fish need to persist in the environment, are they able to access the water and the river and habitat that they need in order to survive? And then this thrown on top of that is through climate change and severe droughts, severe abstractions, you get a river that should be flowing that is now no longer flowing. And that brings in the concept of environmental flow. So not just in-stream structures, but if a river stops flowing, that's a huge hindrance and can be very detrimental to the movement of fish in a system. And this one's always a good picture because it demonstrates what a river should not look like. And this is basically the bottom of a river that should have water in it. So again, if the picture on the right is the weir, and what we try to do is create structures in order to allow fish to move in a system. And you can see the top right here, there's what some form of a structure that's been put in place to try allow fish to move upstream. And then you've got the weir wall itself, and generally the fish can't move up here, but they should be able to move up there. But this is where fish behavior is really important, because we've built this thinking that fish would find this and move up it, not a problem. But if I were to show you this video, this is often what then does happen. Is that the fish will come, and this is the same way in the picture on the right, but they'll come in and actually find and try to push upstream outside of the structure that's actually being designed for them. And so there's a disconnect between what we're designing and what the fish require in order to move upstream and over some of these in-stream structures. So just to take a bit of a step back again is to look at, well, why is fish important? Lynette, thanks for the introduction again. And what you mentioned was fish are often seen as food on the table. And not only that, but there's also an aesthetic value to it through the recreational fishing. And we've been able to quantify that in South Africa with the recreational fishing angling industry having 1.3 million participants and generate about 32.6 billion rand per year to economic activity and sustains just over 100,000 full-time jobs. And that was based on a study by Potts et al. In 2023. An earlier study just looking at the yellowfish recreational angling industry out of Joburg looked at estimate value of 133 million rand to attach to the yellowfish recreational industry. So there's huge value in protecting our fish species and its contribution to our local economy. Not only that is that inland small-scale fisheries are seen as really important in maintaining livelihoods. So this translates maybe not necessarily in huge economic value, but definitely in livelihoods and maintaining people off the poverty line. People that have access to water will fish for fish and be able to put food on their table and possibly even sell the odd fish here and there that's able to then pay for school fees for their children. And then lastly, the importance of fish is that they're very good ecological indicators as to what freshwater ecosystems are telling us. And this we can see when we have quite a lot of poor water quality in our river systems, and then also that translates to fish kills, which can then not only be detrimental to the environment, but impact the food systems attached to those fish. So just to quickly unpack the different types of movement that fish show or demonstrate is that we get two main categories of movement, diadromous species, and then we get pentametrous species. And we'll start with diametrous species, where these are fish that move between our river environment through the estuary into the ocean, and then back through the estuary back into the river. And the type of movement, diadromous type of movements, there's three different types. And we'll go through these quickly so that people can understand what the different types of fish migratory movements are. So anadromous species are fish that will breed in fresh water and then hatch and move back down through the estuary into the marine environment and live in the marine environment to come back through the estuary into the freshwater environment to spawn and complete their life cycle. And some of those fish will then either die after breeding, or they will return back to the sea and start the life cycle again. In South Africa, though, we don't have too many anadromous species. One species that we do have that's actually an introduced species is trout. In their native range, they're considered anadromous, but they also can complete their life cycle without having to return to the ocean, which is the case in South Africa. Another important type of diadromous movement is catadromous species, and these are now the reverse to anadromous species. So they start their lives in the marine environment, hatch, they will drift or move into estuaries, into the freshwater environment where they'll live the majority of their lives, and they move back through the estuaries into the marine environment to spawn and sometimes die. And here there's three really important species of angulate eels, which are the freshwater eels that live in our waters in South Africa, and they can grow really big and often attached to a lot of cultural and local beliefs associated with the species. That if you were to break this migratory route and prevent these fish from moving in and out, you also start to lose the cultural ties associated with these species. There's a few other non-angulate species like the flagtail at the bottom that also have a similar catadromous lifestyle. Then lastly is amphidromus. Here, it's fish will live in fresh water, breed in the estuary. Larvae might drift out to the marine environment, and then the fry come back into the estuary and then move into the fresh water. So it's more of a it's less of a distinct in and out, but more of a existence in the three different aquatic environments. And here we've got quite a few different species associated with Amphidromus species. The important, well, popular or known one would be the gobby species that occur in our rivers. And then the other major movement type is the Ptamidris species. And these species don't require having to go out into the marine environment to complete their life cycle. So here you'll find that they will just need to move upstream and downstream, and also just often just need to find the habitat that they need within the system to spawn or um, or to feed or to even seek refuge during low flow periods. A lot of the species in South Africa freshwater environments partake in these type of migratory movements. So you'll see the tilapia, the cyprinids, which are the yellowfish, and the minnows, as well as the large catfish or barbel that many people may have seen on a plate or eaten some of these. So come back to the importance of fish behavior, less from a economic perspective, but why it's important for us to know is because how fish behavior links into the system. So fish behavior is, well, there's various ways to measure biological organization in terms of we can look at how a fish responds at the suborganism level using biomarkers and bioassays. And then we can also look at the ecological relevance of the system, where we're looking at fish health, population structures, bioindices, and communities. But fish behavior is quite uniquely placed in that it sits right in the middle, where the sensitivity of response meets the ecological relevance. So what happens, how the species responds to the environment happens in real time through the behavior of the fish itself. And so that's what makes it quite important and relevant in monitoring freshwater ecosystems, is that it's a direct response as to what's happening in the system at the time. So just quickly, the use of technology to obtain fish behavioral data is growing, and this is fish telemetry, or often known as fish telemetry. And fish telemetry has been used since the 1950s, possibly a little bit earlier. In Africa, it only really started to be applied in the 1980s, but it's quickly becoming globally the preferred method for understanding fish behavior. And because of this, is we can look where we can't see. So attaching electronic devices to fish allow us to know where fish are moving, how individual fish are responding to different stresses in different environments, and so on. And it's been well developed for inland waters over the years, and particularly in the temperate regions. And here the two main technologies used are radio telemetry and acoustic telemetry. And to explain that briefly, is acoustic telemetry is when we attach a sensor to a fish or a tag to a fish that will give it that fish's locality, the signal is transferred within the water column. So on the right here, you can see the tag will give off a signal that then is received by a receiver that would have to be in the water to be able to give us the locality of that fish. And the locality is important because it tells us where the fish is, what it's doing, and where it's going, what habitat it's utilizing, etc. With radio telemetry, the tag is placed on the fish, but the receiver is placed outside the water column. And so the signal sent from the tag to the receiver, and all that is the tag on the fish sends out a signal to a receiver, very much like a radio station sends out a song, and that we can pick it up on our receiver, which is our radio. And so that we put in an electronic device onto a fish that has the same principle. And we walk around with the receivers to then find out where that signal's coming from. And we can triangulate to see where the fish is. So there's lots of different limitations around these technologies, but what's really becoming evident is the radio telemetry is more applicable to our waters, particularly inland freshwater, where the environments aren't conducive to the use of acoustic telemetry for a number of reasons. This is just a breakdown of all the telemetry studies that have taken place over since the 1980s, when the first few studies were applied on tilapia. And then you can see a steady growth over the years to a lot of the work that we're doing today coming out in more recent years. It's still underapplied in the region compared to globally, and radio telemetry still dominates the inland freshwater studies. And what's emerging now, which we'll get to later in our talk, is a passive integrated transponder technology, where different to the radio and acoustic telemetry, but is very much like the chips that we put on into our pets that mark that pet and is read by an electronic reader. And we can apply that same technology within the freshwater system with the use on fish. So Fish Track, Southern African Inland Fish Tracking Program. This is a program that we developed locally in South Africa because of the need to really, a lot of these technologies are quite expensive and bringing them into the country is costly. And so we started to look at how we could locally develop this technology along with a company called Wireless Wildlife. And we've been developing this since about 2008. And here we make the use of smart tags with sensors on it. And like I mentioned earlier, the tag sits on the fish, and you can see that here. And this tag then talks to a receiver that's outside the water. And that receiver can tell us that the fish is in that region. And then we use, which is slightly different and could be quite technical, but we use ultra high frequencies to communicate the date and position of the fish. So just short on Bluetooth frequency ranges, we're looking at this tag talking to the receiver using ultra high frequencies. And then because of this, we could actually do both a remote method of monitoring fish as well as a manually monitoring fish. So manually, your researcher would go out and look exactly where the fish is, whereas the remote stuff would be the receiver on the right here would then detect when the fish is in range of that receiver, and we know that that fish is in that body of water. And then we could also change the different rates at which we want to communicate with the tag. And we could also store data on the tag, things like temperature and activity counts of that fish. And that started to open up a lot more doors in what we could do with behavioral data coming from fish. And then what we also did was we incorporated a water quality component, which fed through the same system, that these sensors that we deployed into the environment could also detect water quality and flows in addition to just the fish behavioral data. So this is the fish track program, basically a synopsis of what I've just explained. We've got the tags and sensors deployed into the system that then speak to the different stations that then go into a real-time data management system. So not only are they collecting the data, that data is being pushed into the cloud, where then us as researchers are able to access that data in real time and then start to make management decisions based on how fish are responding in the system. It also answers a lot of important behavioral and biology and ecology questions that can help management and conservation of the fish species. And it still incorporates traditional manual monitoring techniques where we can go, if a fish isn't calling in for a while, we can go and have a look and see where that fish is and why. And if it moves out of range, we can go out and find it. So it's very versatile and robust for our systems. So just a quick where we've come from, because it's important to understand that, is that we've taken about 8 different case studies across South Africa to develop this fish track. And I'm just going to highlight three of them that really were a game changer in the way that we could monitor fish behavior. So the first one was similar to acoustic telemetry, but the remote component of it. So we could set up arrays across the river, and as a fish moved in and out of this dam, for example, we could detect how far upstream it moved, which habitats it frequented, and when it moved back down into the system. And that's kind of using the remote system as a gate system to determine the movement up and downstream in the river. Then what we were finding is with the data storage is that we could only get data when the fish was in range of a receiver, but we wanted that data to continue when we couldn't detect the fish, particularly around temperature and data. And so what we started to do is, because these are smart tags, we could store data on the tag. And when that fish came back in range, it could dump all the data back, and we could determine what its activity was during its period outside of detection. And so instead of having big gaps in our data like this, we would then actually be able to fill the data when the fish moved back in range of the station. And then just integration of water quality parameters into the system was really important, because that helped us understand how fish can then respond to different environmental elements and exposures in the system. So particularly discharge flows, as well as the temperature and electrical conductivity. And so this was also developed part of the probes, and there's some of the raw data at the top here of the temperature and the discharge, and what that means to change in behavior, change in habitat in the system in terms of availability for fish. So just some results of this. This was quite helpful in that we could actually show that fish behave differently in different times of the years, as well as different habitat types, and when they moved in and out of their refugia habitat over the seasons. And it really shows that actually fish also need time to rest in the evenings, and then they would move up and move around feeding during the day, and then go back and have a period of rest in the evenings again, often so showing distinct deal patterns in their behavior. The temperature was similar in that we could show that when we monitor the temperature of the water, fish actually still find microhabitats in the system in order to maintain and regulate their temperature as exposed within that system. And that's important as fish are ectothermic, and so they respond quite rapidly to changes in temperature. And then we could also do some of the analysis and the responses as to different temperature, different water quality components, and how fish respond to that. And then just what was also interesting is we could also start to detect specific events. And this is always an interesting one, because so the blue line is the temperature that fluctuates daily, and then the green line is the activity, which also has that daily fluctuation of the deal patterns of the fish. But then we've got this big spike in behavior here that tells us something's happened in the system. And this is what we were getting to with the real-time remote monitoring component. But you also see that the temperatures now starting to reflect atmospheric temperature, not water temperature. And so at this point here, we realize something's happened, and what's caused it, and why has it done it. In this case, it was a fish eagle that had taken the fish out the water and eaten it. But this could also be a pollution event or anything that causes a major change in the behavior of the fish that we could detect through the activity reading. And so this particular event was that we actually showed that as the water levels dried up in a river, tigerfish being very sensitive to oxygen changes, were gulping at the surface and became more susceptible to predation by fish eagles. And so we could link that behavior back to the importance of providing environmental flows. And this comes then down to the next point here, the environmental flows. While we're monitoring our fish in partnership with the Ngeni Utugela Water, we asked them to provide environmental flow release to see what our fish would do. And we could show them that when they released the fish, there was an initial spike in behavior of the fish, which is here in green. The environmental flow is in blue, and the temperature of the water is in red. And we could get all this information together that really helped us to understand what the system was doing. And what we realized was actually that sometimes these environmental flows, as much as they are to help the environment could actually also be quite disruptive in determined in the rate at which they released. And this is an environmental flow here, and then the rains came a little bit later, and you had more of a natural increase in environmental flows. So it starts to ask a lot of the questions of how we should be releasing environmental flows and trying to mimic natural conditions. So with that, we started to develop an alert system to say, okay, well, when there's a big change like this, what's happening in the system? And we really just took a univariate approach where we looked at what the fish's behavior was to predict what it should be going forward. And then we included its behavior back into it to see what its current behavior is to its predicted behavior. And whether there's a significant difference between that would determine whether there's an alert or there's no alert or something to worry about in the system or not. We're starting to look now at the multivariate stuff in terms of trying to not just look at a univariate system, but to integrate other environmental parameters into the system. And this model's been developed and the algorithm's already running. And so it starts to show you here when there's an importance in terms of what's happening. And these icons at the bottom show you that, okay, there's a high alert. In other words, the degree of change is very high and shouldn't be. And the yellow is low, and there's a change, but it may not be significant to what we need or looking at in terms of management. So then we applying this in two catchments at the moment. One is across a transboundary in between Mozambique, Eswatini, and South Africa on the Komati River Basin. And that's really to try and improve the way that we manage transboundary waters for the ecological component. And we're starting to move into a management user interface. A lot of what I've explained is very technical and can get lost in a lot of the managers that really just need a snippet of information to make a decision. And so we're looking to see, okay, well, how does this look? If we can run it in real time, that managers can have access to the platform that we can look at. Yeah, and then this is also then relates to the ecological reserve and what it needs to look like and how fish are responding in providing those flows. And then we're also looking at it using FishTrack from managing recreational fisheries linked to national water monitoring. Here we're looking at regularly tagging 5 fish per year to see how they move in and out of the study area between recreational anglers and subsistence anglers. And then we're also coupling that with mark recapture to see if we can get an idea of what the population's doing. And this idea is this to feed into the kind of FTON program where we can actually use fish behavior to understand what our water resources is doing, as well as improving our fisheries for that reach. And then just we've been tracking what's nice, the versatility of this program is that we can include semi-aquatic species. So we've done some terrapin work recently. We've done some semi-aquatic species tracking. And we've also done some crocodile tracking using the same network approach, the FishTrack program. Okay, so that's the FishTrack program, and I'm not sure if there's any kind of questions, Lynette, that we want to address, but maybe with sake of time, we move on and take questions at
Yeah,
the end.
we don't have any questions on the chat, so I just want to remind our listeners or our attendees today that you can drop a question on the chat, or at the end we might, if we have time, we might be able to take verbal questions as well. So I think, Dr. Vanette, maybe we can just move on and check for questions again later.
Great.
Can I also just take the opportunity quickly to ask people not to forget to register? There's also a link on the chat to register your participation.
Thanks, Lynette. Yeah, so carrying on then is how we look at fish moving across some in-stream barriers or weirs and stuff that we built in the river and that cause disconnectivity in the river. And so in some cases, this is important, like you can see on the bottom right, where we want to stop the movement of fish. And the other one is where we've hindered the movement and we want to now restore the movement of fish. And that's kind of the tale of these two weir types, is that sometimes we don't want fish to move, but other times it's important that they do. So the first one is a study that we did where it's kind of less of two evils, where in protecting the trout industry, which was established in the 1890s, and there's been backed by ordinances, and really to kind of protect the species in linked to the recreational angling. Although we've learned a lot since that and realized that we need a more holistic way of managing the trout, is that they are threatened by other invasive species like bass. And bass was introduced a lot later than trout, didn't have such a strict introductory program because they took to our waters a lot easier. And initially, when they were introduced, we didn't consider the invasive potential and threat to biodiversity. But this was a study where again, to provide water for the region would have inundated the Inchabraki waterfall. And this waterfall actually protects the upstream waters where trout occur from bass. And bass can, particularly smallmouth bass, have a detrimental effect on trout. And so when the dam was built, this waterfall that you see here would have been inundated by the impoundment back in the dam. And so they had to create a natural impoundment or a weir to stop the movement of bass upstream to mitigate the loss of this natural barrier. And for that, they built the Inchabraki weir to replace the Inchabraki waterfall, and was really to protect the upstream trout from downstream bass moving in. And we just did a study there where we looked at what's the species distribution probability if fish do get over the weir, how much of the trout waters that they would infest that could actually threaten the trout recreational angling. So that's a case where we need to protect not only trout waters, but sometimes important species from bass. But then in some cases, when we put in barriers, we need to get the fish over the weirs and moving through the system. And this can be a lot harder, stopping them from moving upstream. Or it's a lot easier to stop them from moving upstream. It's a lot harder to get them to move upstream. And so this often requires what we call a fishway or fish ladder. And according to the South African Water Act of 1998, it requires that river connectivity is maintained in the development of our infrastructure. In-stream weirs in South Africa make use of a vertical slot fishway design. And this is now considered a standard in abstraction weirs for South Africa. So particularly a couple of ones that have been built here in KZN have got a vertical slot fishway in it. However, the efficacy and the total pass of the vertical slot fishway is still to be fully determined in its real-time application. And so this is where some of the studies that we've been doing have been a first for the region. So one of the problems with dams, as many others, is that they do obstruct the obstruct the movement of fish upstream, and we need to really understand how fish behave in order to help them move up over that barrier. And the standard one was mentioned earlier as a typical vertical slot fishway, but fish can't read signs, so it's very difficult for them. They read cues in the environment in which to access the mitigation measures that we've done. And so vertical slot fishway is really just simple terms is a step ladder for fish to come up over the weir and around it to maintain their movement through the system. So the study we did on the Tugela River fishway, this is a picture of the weir, and you can see the barrier that it creates. This is a rock ramp that's designed to help with the macro invertebrates, the creepy crawlies, to get up over. And then this is the entrance to the fishway here. And you can see a lot of debris, and the fishway actually goes up the wall here, up through there, and then through the obstruction bay upstream of the weir. So the fish actually have to come in, find this entrance to move up. And so this is just basic design of the vertical slot fishway. This is the entrance of the fishway moving up the two dog legs and then out into the top there. This is the actual drawing in the middle here. And then these are the baffles that help slow down the water and create a gradient in which the fish can move up. For this study, we used the passive integrated transponders, and this is a 2-gram, 12 millimeter long tag that we then eject into a fish. And this was a fish that we recaptured. And because the gobbies, freshwater gobbies actually are quite translucent, we could actually see where we had injected the tag, and we could see that it was healing nicely and there was no impact on the fish. And that's what you can see there. And then we would scan the fish, and you could actually then get an ID on that fish, and that this tag then helps us know that this fish is in the system, and we're able to detect it. So you get a handheld scanner that helps us to determine that, but then we built these massive big antennas that then get slotted into the fishway. And when the tagged fish move up through this in the fishway, we can then detect the fish. So this particular antenna was placed at the top of the fishway, what we call the upstream exit. All the fish were tagged downstream, and what we're looking for is total movement of fish, detecting through the fishway. So total pass efficacy on this system is we were only able to tag 107 fish, representing 12 species, 8 tagged individuals representing 3 species successfully navigated the fishway. So out of the 107, only 8 species, 8 fish were able to move, and this kind of gives you a total efficacy of 7.9%. Just to show you, we had the highest pass of catfish. The cyprinids were not as efficient, although they were quite low in terms of our numbers tagged. And then Mozambique tilapia we had one that was actually living in the fishway that we collected a few times. And then this is a new fishway that's just been built. The water started moving through the fishway this year, and I was in the field last year installing our monitoring device on the fishway. But it's a similar fishway to the Tugela ones just explained. Vertical slots get put in, rough base to help with dissipation of flows, and then the fish would move up through the structure to get upstream. And then this is the flow of the water coming downstream, and the fish will then have to come up into the system to go up and around. For monitoring, we actually fortunate to have 4 monitoring devices on this system where we're able to really test the efficacy of fish coming in, because the fish has to find the entrance before it's able to scale it. And so finding the entrance becomes really important. If they're not finding it, then it may deem the fishway redundant. And then so the fishway setup is 1 in the entrance, 1 just 3 baffles upstream after the first turning pool, and then total pass to see how many fish are able to find it and actually then scale up to the top. So these are the monitoring devices going in, and then you can see them coming in here. This is our first one, our second one, and then our third one's up there, and then you can just see the top of the box that covers the exit. So the efficacy of the fishway entrance, this project is ongoing. It's still early, so there's pictures are hot off the press. But we've tagged around 53 different fish to really determine if the fish can find the fishway, and then can they move up and pass through the fishway. And then once through, what's intriguing is that they still have to navigate the abstraction weir pool before, or extraction well before entering the weir pool and then moving up. And the question always is, can we better design the fishway? In many ways, we probably could, but it's measuring the efficacy of what is standard practice today to see how we can measure them better. So just future direction for fish telemetry, we're looking to prioritize movement behavior or fish behavior into aquatic management, direct actions to the proactive management of ecosystems using fish behavior, the adoption of fish telemetry into freshwater monitoring programs, and you can see that's a lot of the work that we've been doing, understanding the impact of in-stream structures on fish movement, improve infrastructure around real-time monitoring of aquatic systems, not just fish telemetry, but even other chemistry parameters and flows. And then validation of the efficacy of built infrastructure catered for river connectivity. How well are these fishways working? And then try build capacity and simplify technologies for wider application in Southern Africa. And then also kind of really trying to look towards using fish behavior-driven management practices. So let the fish tell us how effective we are rather than trying to tell the fish what to do. So thank you very much, Lynette and the group. This is just a couple of photos from my students over the years and some of the pictures of the fish that they've managed to capture themselves. So thanks very much, and thanks everyone for listening.
Thanks so much, Dr. Matt. I think I've learned a great deal about fish that I didn't know. Um, we don't really have any questions on the group, but we did have from Matthew Ross. He is also involved in the creation of fishways, designing of fishways, it looks like. And he is requesting if we can get in touch with you, see how the work can align with his. Would it be okay for me to drop your email address on the chat?
Yeah, please do. Thanks, Lynette. Hi, Matthew. It's good that you joined us today. And yeah, good to see you on the call. And I think you've got a good update as to where we are now.
Yeah, and I'm sure, it was even more helpful for some of our other viewers today than it was for me. For me, it was completely learning. But one thing
Yes.
I found, that immediately came into my head was when research is being done, what is the most surprising thing that your data has actually over the years shown you? Anything weird or, something you hadn't anticipated or was counterintuitive to how you thought the fish might be behaving? It's just the most interesting thing you've come across.
Yeah, that's always a good question to ask. I think with the telemetry stuff that we do, the fish take us to places that we probably least expect. And so some of the interesting experiences that probably don't come through the research as much and only through the presentations is that when I was tracking tiger fish in the Kruger National Park as part of my postgraduate studies, we did a night survey on the fish, and the tiger fish was actually sleeping in the backwater. And so we were able to track it, and I could see the tag on the fish. It would sleep at the surface, and then when I shone the light on it, it would just drift down into the deeper water and then come back up. And I think that just shows to me was really a aha moment in the sense that this technology really works, and we can really refine the fish that we've caught using it. And that's just really, we often forget that animals and fish need the same basic needs as us people in terms of that. So they need to rest, they need to feed, they need other fish to live with and breed and so on. And so that I don't think was unrealized, but it just became more kind of more realized in that way that fish they're complex beings similar to us, and they need a multiple array of different habitats and stuff to survive. I think on that one other one was I was trying to find a tiger fish and I couldn't find him, and so I put the antenna on my shoulder, and then I could hear the signal coming, and I'm like, that's very weird. A fish shouldn't be out the water. Um, so we went off to track it and got charged by a line in the process because we were in Kruger.
Oh, wow.
And but we found it in a fish eagle's nest. And so that just also highlighted, that's not, again, not what we expect to find in our study, but it is, we knew it happens. So it's just a really cool to validate that type of data through the work that we were doing. So yeah, we found the fish tag in a fish eagle's nest.
Oh, wow.
So
You
that
might
was
even have been tracking a fish eagle instead of a fish if it had
yes, yeah.
followed it.
So,
Oh,
so
that's
that's
interesting.
where yeah, so that's where that obviously if it's flying around, it's not a fish anymore.
It's not a fish. And then
Yeah.
the other thing that came to mind as well was, would you need to do this sort of study? Let's say somebody wanted you to monitor fish in another river. Would you have to replicate the study every time, or are there certain things that you can sort of almost generalize, or it would be environment specific?
Yeah, so I think some of the like the deal behavioral patterns and I think species you could be transferable to different systems. So if it's the same species, you expect them to behave in the same way. Different species will behave slightly different. And you know, we've come a long way, but we still don't know where for example, the yellowfish in a natural system that aren't tied to a dam are moving in between seasons. So each different system will have slight nuances that would need to be discovered. But we've done a lot in terms of the baseline work to understand, okay, well, most of the fish that we've studied would exert deal behavior. And it's those slight nuances that taking to a new system would help us with. So similarly with the fish way, generally that is a standard engineering design, but different species might behave differently approaching it, or the fish way is built differently. So although the Tugela and the Kumar system are the same, the fish way entrances are slightly different. And that could actually make or break the system. And that's kind of where it's important to validate and measure that so that we know that that system's working as efficiently as we need it to.
Okay, great. So it means there's a lot more scope for your work.
Yes, yes. And I
A
think
lot more to learn.
yeah, and it's exciting, you know, like I was saying, it's exciting work to do. Um, and we actually need to be doing more of it. And it's trying to find that capacity and capability to be able to roll it out so that we're not the only research group doing it, but there's other people prioritizing it and investing in it. So yeah.
Yeah, I think it's that investment as well, where even webinars like this is very good to just highlight something,
Mhm.
highlight the importance of something. Dr. Bennett, thank you so much for your time, and for you know when we approached you for National Science Month, for being willing to come and tell us about your research. And we hope that there will be other opportunities like this, and we don't have to wait for National Science Month next year, that we
Yes.
can have you on for some other platform again sometime
Yeah,
soon.
great. Thanks,
And
Lynette.
also then thank you to everybody that has joined us this morning. As I said, I learned a great deal this morning, which is always good. We also hope that people have joined any of our other activities for National Science Month. It has been quite a roller coaster for us, having been our first full month length National Science Month, and we look forward to next year. We've learned a lot. And we'd also like to encourage when you are registering for this event, for example, on our same system, if there is a space for comments, if you can also just let us know, how we've done today, if you found the webinar useful, or if there's anything that we can improve for ongoing forward. So thank you very much for your time, and thanks to the team on the back end, Kenneth and Sipo, for the assistance this morning. Thank you very much. Enjoy the rest of your day.
Yeah, thanks, Lynette, for everything. I'm gonna log out now. Thanks again for hosting this.
Thank you so much for your time.